Hello, guys. I hope all is well. And the next chapter, my pen, I just had it here. I'll be done. Next chapter, is chapter on pause recording. Sorry about that, guys. Okay. So this chapter is over the DNA technology, what they are doing nowadays with DNA. And then I hope you read the textbook, you understand the PowerPoints and everything else. Let's start and see what happens. And if you have any question, please let me know. So your textbook saying they found they were able to complete two of the species based on the DNA, the small amount of DNA that is left over from Neanderthal man and woolly mammoth. They have been able to complete the genome of these two organisms. And then once in the radio while I was driving, they were talking to a geneticist, and then he said, it's amazing the progress we have made in last 10 or 20 decades. Before we never thought of it, that we are gonna make that much of a progress. And then you will see some of these that is happening. Okay. So biotechnology, make sure you know the difference between biotechnology and genetic engineering. Biotechnology is the manipulation of organisms or their components to make a useful products. For example, they use corns or they use animals. If you know an animal gives a lot of milk, so you cross it, you breed it with another animal that gives a lot of milk of the same species. You know what I'm talking about, like cows. So that is a biotechnology. So biotechnology have been around for thousands of years. It's same as crop and some of the things that Mendel proposed. Okay, that was, if you cross this flower with that flower, you get perfect flower, and then you cross purple flower, you get this and that. So that's kind of biotechnology. Now, what is nowadays is genetic engineering. We are able to manipulate the elements that Mendel was talking about. So the genes that we can manipulate them and change them and make corrections. You will see it today. That is a genetic engineering, direct manipulation of genes for practical purposes. And what is DNA technology? The applications of DNA affect everything from agriculture to criminal laws to medical research. All of that is DNA technology. That's what this chapter is all about. So let's say if you want to, for example, your textbook is saying, if you want to make a growth hormone for a human, or you want to make a crop that gives a lot of yields or resistant to something or anything, heart medications, proteins for heart, anything like that. So these are the protein molecules that are made inside of our cells, like insulin. I'm going to write down insulin, growth hormone, okay? And that is only and only in human made. Of course, animals have their own insulin, like pigs' insulin is very similar to human insulin. In old days, somebody who needed insulin, they took pig's insulin from the pancreas, and it was very hard, laborious work. They had a lot of money, a lot of pigs to collect a little bit of insulin, and only rich people could afford it. Nowadays, anybody who needs insulin, easy, very easy, and exactly like human insulin. Pig's insulin is a little bit different than human insulin, and some people could have reaction to it because it was foreign to human body. But nowadays, insulin that bacteria make are exactly the same. So let's say how they make it. What they've done, what they're doing, they take the gene, here is a gene from an eukaryotic cell, that type of cell. And then they take the plasmid out of the bacteria. Plasmid is circular DNA. They take it out. And then with the help of restriction enzyme is able to break open the plasmid, okay? And you have sticky ends. Remember that? So it breaks it open, And then with help of DNA ligase, they take the gene for insulin or growth hormone or anything else. They attach it to the plasmid. Now they take that plasmid and put it in the bacteria. And guess what? Bacteria uses ribosomes, uses all this machinery, and it starts making insulin right here. So that's how it works. Okay, so you need two enzymes. Of course, there are more steps involved, but pretty much for what I'm talking about, you need the restriction enzyme, you need DNA ligase. And of course, you need a bacteria, and you need the gene from eukaryotic cells that is your desire. Here they are. They're showing it to you in more details. And DNA chromosomes, they were able to take out the gene that you want to, and then they put it in a plasmid, and then you let the bacteria grow, and then you have many genes. Remember, you started out with one, right? And then now you let the bacteria grow, you have 2 or 3 or 4 or 5 or 10 or 100 or thousands, okay? Right here, you have more of it, more bacteria. And then of course, the bacteria will start making the insulin or growth hormone, what have you. So making multiple copies of a gene or other DNA segments to work directly with specific genes, scientists prepare well-defined DNA segments in multiple identical copies by a process called DNA cloning, which I talked about that. That is, you also multiply, when the bacteria multiplies, of course, the DNA of the desired gene that you put in the plasmid, that is also multiplied, okay? So plasmids are small circular DNA molecules that replicate separately from the bacterial chromosomes. So the researchers can insert DNA into plasmid, into, okay, I talked about all of this. Molecule with DNA with 2 different sources, and then, you know, I just talked about it. Let's move on. Using restriction enzymes. So for the lab, you're seeing what is happening with restriction enzymes. Restriction enzymes, bacteria use it to defend themselves against viruses, the bacteriophage. So restriction enzymes breaks down DNA. They recognize its sequence of AT to AT to T to A, T to A, T to A, or A to T, so on and so forth, and they break it, okay? A restriction enzyme usually makes cuts yielding restriction segments, okay? And then the end of these ends, if this is a plasmid, it breaks down to this and that. So that end, these ends are called sticky ends. And sticky ends can bind with complementary sticky ends of another fragment, which I'll show you sometimes. And DNA ligase, of course, comes together and seals them together. Here he is. So in this diagram, I should talk about restriction enzymes. I should talk about sticky end DNA ligase. Okay, great. So here is it's a plasmid right here. The word that are bacterial plasmid, and plasmid is only found in eukaryotic cells. In prokaryotic cells like bacteria. They are not found in eukaryotic cells. Your cells and my cells do not have plasmid. Okay, anyhow, having said that, so the restriction enzymes recognize this right here, A to T, and breaks it up. And here is sticky end right here, and here is that sticky end. So you can use the desired gene. For example, this is insulin, right? The gene they got it from human chromosomes. They take that one and they insert it here. And of course, DNA, so that's called sticky end. This is restriction enzymes, break this, okay? And then what happens, you have sticky ends here also, right? And then what happens, DNA ligase put these together. Put this one, the purple one, into the green one, okay? And now at the end, you have this segment. Okay, that's a recombinant DNA molecule. It is not same as the parental DNA, original DNA. This is the recombinant DNA. You combine something else to it. Oh, video, let's watch it.
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences of DNA and then cut the DNA to produce fragments called restriction fragment. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
Okay, so these are all of different type of restriction enzymes, and they all have a name. If you look at the restriction enzyme for lab, it says R0H21, something like that, some strange names. All of that is the name of the restriction enzymes, and they got it from bacteria, where was the source, and so on and so forth. So they give a nomenclature. To take the recombinant strand, researchers might use the gel electrophoresis. So if you cut a DNA molecule, for example, I'm gonna cut this. If you have a DNA molecule like this and you cut it, so you wanna see, have you cut it or not? You cannot even see it on the microscope. You have cut this DNA or not. So what they do, they use a system called gel electrophoresis. They load it up, you've seen it in the lab, into the well. This is a gel right here. This is a gel right here, okay? And these are the wells. You place them in the gel. These are the wells right here. And then you put the DNA samples right here, right here, right here, right here, and you pass electricity through it. The DNA is negatively charged. So when you put them up here, the DNA is negatively charged. DNA is negative. If they move toward positive end. So they are moving. The larger fragment stays up here, and the thinest one, lightest one are here. That's light, heavy. Okay, same thing here. So it shows, this shows that restriction enzyme worked. So the DNA was cut to pieces. The DNA was not like a long strand like this, okay, like that. So DNA was cut to pieces like that. That shows, that's what the negatively charged DNA moves toward positive electrode. That's what I'm talking about. Okay. So here is another one. Let's look at it. I guess we do not have volume for this one, guys. Here they are, just showing you how they're loading things up. These are automated systems they use in some research like robotics. So, sometimes you have to autoclave it. You have to bring it to 37 Celsius. I think that's what you're doing here. You do not have volume. Okay, that would be possibly cell growth. They're working under the hood to grow cells, and they were doing different things. Okay, guys, that's me. Okay, this is another thing that the biotechnology experiment we have. Make sure you study it. You know everything on this slide. So you have a bacteria that has the gene ampicillin. So this bacteria is resistant. It has a plasmid, and that gene throughout that plasmid make that bacteria resistant to antibiotic ampicillin. Okay, then you have another bacteria that it has a gene on its plasmid which makes that bacteria resistant to another type of antibiotic called kanamycin. And then what the goal of this lab would be that you combine these two plasmid together and make that plasmid, that bacteria with the plasmid inside of it, resistant to both ampicillin and kanamycin. Okay, so what you do, use the restriction enzyme. Right here is the name of restriction enzyme. I gave it to you guys. And then you break it apart. When you break it apart, then use T4 ligase to put them back together, to put the sticky ends of this one and that one together. You, of course, for doing this, you pass the fragmented DNA through gel electrophoresis. After you've done that, you use the T4 and you know the experiment work, your restriction enzymes work, separate them. Then you put these two fragments together and you have a plasmid that has both genes for both antibiotic resistance. And you put both antibiotic on the bacteria will not die. On this bacteria right here, if you put kanamycin, the bacteria will die. On this bacteria, you put ampicillin, this bacteria will die. But on this bacteria right here, you put both of them, kanamycin and ampicillin, it will not die. Okay, I hope I made some sense. Just the experimental purposes for experiments. And now they have a guideline for researchers that make sure you use the right type of ethical honesty. Then back in 1993, 94, somewhere in that neighborhood, this one guy, I forgot his name. I should have put his name down. He was from Berkeley, right around our neck of the wood. And he won the Nobel Prize all by himself. If you look at the recent history of Nobel Prize, you will see in science or in chemistry or biology, physiology, you will see a group of 2 or 3 or 4 scientists win the Nobel Prize. This guy won it all by himself, okay? And he went to school in Berkeley. But anyhow, so he came up with the idea of polymerase chain reaction. You probably have heard about it in news a lot, PCR. A lot of laboratories around the world depend on this technique, okay? So, and this technique has 3 steps. I would like you to know that. Heating, cooling, and replications, which the name for them is not heating, cooling, and replications. It brings about the chain of the reactions. No, it's called, it will come up, okay? So right here, you have the original DNA, this one. You heat it up. Of course, it denatures, and when you heat up the DNA, it will separate. Annealing, okay, denaturation, your textbook saying denaturation, okay, well, that's fine. Annealing and extension. So there are 3 steps that I was trying to say it in the last PowerPoint right here. Your heating, cooling, replications, the actual name is denaturation, annealing, and extension. What happens during denaturation? DNA has 2 strands. It will separate when you heat up the machine. He went to the laboratory and he heated up the machine and then he cooled it off. And then he came back, heated up again and cooled it off and heated up again and cooled it off. So what happens During heating, denaturation happens, the two strands separate. And then there is a primer, the RNA primer right here. These are the primer, as you learned in previous lectures. So that is the annealing happens. So and there are bits and pieces of DNA in the solution. So what happens, those pieces and bits are brought and being put together next to the template. Now you have two strands of DNA, right? The complete strands. And then you do this again, you heat it up again, you cool it off again. It happens, you started with one strand, now you have two. Now from you start with two, you have four. If you start with four, you have 8. So the exponential growth rate is like this. Okay, so that's what happened, and he won the Nobel Prize for it. Okay, techniques, denaturation, annealing, and then of course extension, and then you repeat it again and again and again and again. Soon you have many, many, many copies of the RNA. PCR amplifies DNA. So you started with one copy. Errors occasionally incorporate errors in the amplified strands, so it cannot substitute for gene cloning in a cell. Gene cloning in cells, that is when you took the plasmid out. You remember I talked about it, and then you put the human gene attached to the plasmid. That's what he's talking about. Okay, PCR primers can be digested to include restriction sites, allow products to be cloned into plasmid vectors. The resulting clones are sequenced and error-free inserted into selected. Okay, here segmented fragments. You can make many copies of the gene that you want, and then eventually put them in plasmid and grow them. That's another thing. And then he's talking about a few other things there. Let's see what's this.
We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid. The ampicillin in the medium limits growth only to plasmid containing cells. But which cells have plasmids with a human gene? Bacteria without human genes have intact LAC-Z genes. This allows them to break down a substance in the medium that stains their colonies blue. Thus, the bacteria with human genes are in the colorless colonies.
Right, that's another technique they use. They use coloring and so on and so forth. Okay, but anyhow. So cloned organisms and stem cells, we're getting away a little bit from cloned just genes. Now we're talking about the whole entire organisms or cells of my bodies can be cloned. That's the next thing to talk about. So organismal cloning produces one or more organisms genetically identical to the parents and donated a single cell. The stem cell is relatively unspecialized cell that can reproduce itself indefinitely or under certain conditions. In 1997, I remember that, the Scottish scientists, they cloned this first mammal before other animals were cloned, but they were not, none of them were mammals. So this was the first time a mammal was cloned, and then it died at about 6 years later. Usually sheep live about 20, 21 years, 19 years, but they die early because of one of them is telomeres and the other one, the DNA is too condensed and so on and so forth. So this is the technique I would like you to know. So they took the cells from mammary gland of that organism, that sheep, okay? You can see that. They took the mammary cells and then they took the nucleus out of the mammary cells. Remember, the nucleus out of the mammary cells in case of sheep has 48 chromosomes. We human has 46, but let's say sheep has 48, okay? Then they took the egg cells from that sheep, black-headed sheep, and then they removed the nucleus out of the egg of that sheep, okay? Then this empty egg does not have any nucleus. They put that 48 chromosomes, 2N, into that empty egg. They give a shock, electric shock to that egg. The egg starts multiplying. The nucleated, I hope the nucleus from a mammary cells of a different animal, different sheep, not this, they could do it with the same sheep. They just, in this case, they took a different sheep. And then they put the nucleus inside of the egg, empty egg of a different sheep. And then they passed electricity through it. Then that egg, I cannot say fertilized egg, that nucleated egg became 2 cells, 4 cells, 8 cells, 16, 32. Then they took that blastocyst, the early embryo, and they put it inside of the uterus of that sheep, the surrogate, a different sheep. Gestation period of a sheep is 11 weeks. Yeah, I'm digging in here deeply. I don't know how long it is. It's not 9 months. And then the sheep that was born was exact clone of that sheep. Okay, the white-headed sheep. And of course, there are ethical issues. Shall we clone human or not? So here it is. So again, they remove the nucleus of the mammary cells, and that's what they call it, dolly. The scientists were wondering what should they call that sheep right here, this sheep right here. What shall we call it? They call it dolly because of Dolly Parton. And here it is. They took the mammary cells, and then they took the nucleus out of the mammary cells, and put it in that empty egg. And then this sheep that was born by a C-section, it was, of course, died early. It was called dolly. So cloning has been demonstrated in many animals, including after that, other animals have been cloned, and many more. They're just seeing some copycat animals. The first time a copycat was mentioned, it was not exact same color, and the animals do not behave the same way. So if you're taking your dog to be cloned, you might be surprised how come he doesn't act the same way. Okay, they don't. Stem cells, let's continue that. There are, we have cells in our body, like in our bone marrow. There are cells that are called stem cells, and they differentiate, and they become red blood cells and white blood cells. Okay, and white blood cells, we have 4 or 5 different types of them. So we do have stem cells in our body, but our cells can act, other cells, like you take a fat cells and you put it under some conditions, that fat cells can become liver cells. It can become brain cells. So, or take liver cells, it becomes brain cells, muscle cells. Here they are. So you have a stem cell, a cell division become a precursor cell, and precursor cells become other variety of cells. And that's called adult stem cell. So we have two, but there are more, but there are two type of stem cells. I would like you to be familiar with them. One of them is called adult stem cell, and the other one is embryonic stem cell. Embryonic stem cells, you remember if the sperm goes inside of the egg, fertilized, the zygote becomes 2 cells, 4 cells, 8 cells, 16, 32. You have a ball of cells, and they take the cells from here and they grow it to different type of cells, right? It's called embryonic stem cells. If you take adult human cells and put it in a dish and grow it, and then from that dish, you differentiate it to different type of cells, that's called adult stem cells. This is very difficult to achieve to different type of cells. This is much, much easier. Embryonic stem cell is much, much, much easier. The cells easier become liver cells. The cells easier become neurons in the brain. However, if you want to give me the liver cells from an embryonic stem cells, right? My body is going to resist it because they're foreign. But if you take my cells and make liver cells, okay, that is easier. My body is not going to reject them because they were originated from my own cells. I hope I'm making some sense. So a couple of terms I would like you to be familiar with is pluripotent and totipotent. Your textbook did not give the other one, multipotent, but that's okay. So pluripotent, it means that the cells that can become variety of, they can differentiate to a variety of cells, but not all. That's pluripotent means. Totipotent, it means totally. They can, and that's egg. The only examples of totipotent I know are the cells of the egg. The eggs can differentiate to a variety of cells, many, 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 many cells, okay? So that is something I would like you to know. Totipotent cells are like egg cells that can develop into the entire organisms. Pluripotent, they can differentiate into many different type of cells. And the key word here with pluripotent, not all. Okay, gene therapy. What happens with genes is the alteration of the afflicted individual's gene, and then they use gene therapy. So these are some of the techniques they're going to use. The CRISPR gene, there are 4 scientists who discovered this simultaneously, and they are in the court fighting each other over who has the right. I think these guys will win the Nobel Prize someday. I don't know if they win the Nobel Prize. What will happen? Will they take each other to court at that time to say, hey, no, this is my money, or that is my money? I don't know. Well, they're supposed to divide it among themselves. Usually scientists who win it, they divide the money among them. But now they are in court for copyright laws because they say who discovered it first. But anyhow, it's okay. The CRISPR, they call it CRISPR gene, is a clustered, regularly interspaced, short palindromic repeats. That's what CRISPR gene came. These are what they are. They are protein molecules inside of bacteria, which the bacteria use to protect themselves against viruses that they are invade them. Okay, like restriction enzymes, what I talked about. They use that to break the DNA. So what happens? Uses the enzymes Cas9. I guess I will show you a picture of it. Usually, if you Google it or you read a scientific paper, after CRISPR Cas9 comes up. Okay, which identifies the specific nucleotide to be cut using a guided RNA molecule. They use RNA, they call it gRNA. You'll see it guided. That's what gRNA here it is, guided RNA. to protect the bacteria from Cas9, a sequence called PAM, which is not found in bacterial cells, must be placed next to the targeted DNA sequence. And what is a PAM? Is a proto spacer adjacent to motif, which is 2 to 6 DNA pair sequences, about that many, and following the sequences targeted by Cas9 nucleases in CRISPR bacteria adaptive immune system. Let's look at some pictures. Here is the Cas9, the CRISPR Cas9. Here is the DNA, and let's say there is a gene right here that needs to be fixed. Okay, it's giving a person a disease such as sickle cell anemia. Okay, so what happens? This is your RNA right here. They call it guided RNA, gRNA, and it goes through this Cas9 nuclease. It breaks it open. Okay, CRISPR gene. It breaks the DNA open, and based on that RNA, the new strand of DNA is going to be is going to be made based on DNA right here. And of course, the Cas9 and everything comes off, and that gene that was a problem right here, it is fixed now. Okay, here's another picture, CRISPR-Cas9 at the very first step. This is the targeted DNA right here, as the DNA that is malfunctioning, you know, like, as I said, sickle cell line, cystic fibrosis, sickle cell, anything like that. So what happens right here, you have your CRISPR Cas9, it comes through, this is the guided gRNA, right? Guided RNA, and then it cuts it, and then the DNA ligase, it will come and put, after fixing this, DNA ligase comes and put them together, and then that gene is fixed. So here there's a clone gene. You can take a gene. Of course, I didn't say that. You have to have, this is designed, your gRNA is designed based on what is it that you want. You don't want this gene, right? You don't want that gene, but what is it you want? Then you program it. Use the system to take the gRNA and fix it based on gRNA, and DNA is fixed. But anyhow, so they take that clone, that segment, and you put it in a chromosome, you inject it to a person, and hopefully everything goes away. Again, it's not in the market. It's gonna take a while before it becomes, it's in the market. Genetically modified organisms, which we are eating it right now, and there's controversy around that one. GM foods, GM crops, genetically modified. It means they are food that they are resistant to cold weather, okay? But they're resistant to drought, okay? So, but what effect it has on human has not been around long enough to be able to identify, do we have a problem, or, you know, many things, super weeds and so on and so forth. Continues to change in agriculture, industry, and medicine. And that's the end of the chapter. I hope, is that right? Yeah. And I hope you guys learn the material.